Hi, my name is Evie, and for my genetics lab final project, I'm going to be talking about complementation testing. So, complementation testing is a fundamental assay used in genetics to determine if two recessive mutations that produce the same mutant phenotype affect the same gene or different genes. How it works, you can make mutant strains of an organism. In our example, we used yeast. And if two mutations affect the same gene, you would say the strains fail to complement, and the resulting diploid progeny still lack a functional copy of the gene, so they'll display a mutant phenotype. If two mutations affect different genes, the strains complement each other, and the resulting diploid progeny will inherit one functional copy of each gene from each parent, which is sufficient to restore the wild-type phenotype. Today, we will use complementation testing and colony phenotype to determine the genotypes of unknown yeast strains. Before we jump into the complementation test, I'm going to give you a refresher about our yeast experiment. We used a single-celled eukaryote yeast, which are an excellent model organism for their rapid growth, simple life cycle, and easily manipulated genomes. Yeast can either exist in haploid or diploid states, and because they're eukaryotes, they have a nucleus and organelles, making them comparable to humans. In this experiment, we utilized yeast strains designated by their unique haploid mating types, which are MAT-A or MAT-alpha. MAT-A cells can only mate with MAT-alpha cells and vice versa. A key part of this experiment was the adenine biosynthesis pathway. ADE1 and ADE2 are genes that encode enzymes in this pathway, and haploid yeast with a mutation in either of these genes are unable to synthesize adenine, meaning they are adenine auxotrophs, and they require adenine to be externally supplied in their growth medium for them to survive. Additionally, a mutation in ADE1 or ADE2 can cause an accumulation of 5-amino imidazoles ribonucleotide, or AIR, which will result in red colonies rather than the wild-type white color. Another possibility is that if the mutation occurs upstream of ADE1 or ADE2, such as in ADE4, 5, 6, 7, or 8, as shown in this image, the cell still becomes an adenine auxotroph and requires adenine to grow, but AIR never gets the chance to form, so the colony will be the wild-type white color. This is an example of epistasis, where an upstream mutation masks the phenotypic effect of a downstream mutation. It's noteworthy that the frequency of white mutants is higher than the frequency of red mutants because they are revertants and suppressors, and these other upstream genes that can be epistatic to ADE1 and ADE2, and these can all mask the red phenotype. Now, for these strains we used in our experiment, for reference, HBO is the wild-type control, and it is MAT-alpha, and it's a white/cream color. HA1, HA2, HB1, and HB2 are known mutants, and they're all going to have a red/pink color as shown in this image. HA1 is MAT-A with a mutation in ADE1. HA2 is MAT-A with a mutation in ADE2. HB1 is MAT-alpha with a mutation in ADE1, and HB2 is MAT-alpha with a mutation in ADE2. Now for the unknown mutants, XA1 through XA4 either have a mutation in ADE1 or ADE2, which is to be determined by this complementation test, and they are MAT-A. The materials for this experiment. We need a fresh YED agar plate, the known alpha-type strains, the known A-type strains, the unknown A-type strains, and many sterile toothpicks. Now let's jump into the procedure. Firstly, always use sterile technique, spray your gloves and workbench with 70% ethanol just to avoid contamination. Okay, step 1 is to label your YED plate with your name, date, etc. This is something I forget to do sometimes, and it causes a lot of issues later on. Step 2 is, for this complementation test, we are going to draw a grid on the plate as shown here. The alpha-type strains will be across the top, and the A-type strains will be going down the left side. This is looking at the grid from the lid, so when you draw the grid on the bottom of the plate, you'll have to draw it inverted so that when you flip it upwards, it's correctly oriented. Step 3, use a sterile toothpick to streak a small patch of HB1 into its parental slot, shown here. Also streak a small spot, not a patch, of HB1 down its entire column in the 6 test slots. Use minimal yeast. This is very important because if you use too many cells, the individual genotypes won't mix well enough, and you'll see patched diploid and haploid progeny rather than a homogeneous mix of the diploid progeny. To achieve this, tap the colony, don't scoop up a glob of cells. Step 4 is repeat step 3 using a fresh gel toothpick for HB2 in its respective column. Step 5 is to streak a patch of HA1, HA2, and XA1 through XA4 in their respective parental slots, shown here using a fresh gel toothpick. Using another fresh gel toothpick, mix the A-type strains from their parental slots into the alpha-type strains at their intersections. To note, I used the red, purple, and blue just for visualization purposes, but obviously these are not the real colors of the strains, so it's not realistic. Another important note is to mix these very well so that the non-motile yeast cells can physically contact each other to mate. Step 7 is to incubate the plate at 30 degrees Celsius, agar side up, and then refrigerate them. And finally, step 8 is to examine the resulting diploid colonies. Finally, for the analysis. If the resulting colonies are red, the strains failed to complement, so we know their mutations are in the same gene. If the resulting colonies are cream, the strains complemented each other, so the mutations are in different genes. Here's an image of my complementation plate after the incubation period, and to the right are the class interpretations of the phenotypes. Let's go through the results. HB1 crossed with HA2 and HB2 crossed with HA1 yielded white colonies. This was expected because we know their mutations are in different genes, so they complemented and yielded a wild-type white phenotype. Alternatively, HB1 crossed with HA1 and HB2 crossed with HA2 yielded red/pink colonies. This confirms that the mutations are affecting the same genes, so they failed to complement and yielded the mutant phenotype. Based on these data, XA1 is likely to have a mutation in both ADE1 and ADE2 because it failed to complement with both HB1 and HB2, meaning it will have a mutation in the same genes as both HB1 and HB2. XA2 must have a mutation in ADE2 because when crossed with HB2, it failed to complement. When crossed with HB1, the mutations complemented. XA3 and XA4 must have a mutation in ADE1 because they both complemented when crossed with HB1, or they failed to complement when crossed with HB1, and they complemented when crossed with HB2. So here are the overall conclusions for the genotypes. Overall, complementation testing is an efficient way to determine if genes are in the same or different complementation groups, which can help to deduce gene order and gene function. However, complementation testing and phenotypes are just a starting point, not a full conclusion. This means that we cannot deduce with full certainty that the genotypes of XA1 and XA4 are caused by mutations in ADE1 or ADE2. While it is possible, it could also be due to revertants or suppressors or a mutation upstream, like we said in ADE 4 through 8. So a possible follow-up experiment could be to sequence the XA1 to XA4 strains in order to determine with full certainty the true cause of the observed phenotypes. Thank you.